Hey guys, well, we're looking at the genetic and hormonal responses to sprint intervals in elite cyclists. And so the entire purpose of the study is basically we have these elite athletes that are cyclists, and these guys are endurance based, and seeing if we add sprinting into their regimen, how is it going to affect their biochemistry? Are they going to become stronger? Are they going to have a better recovery? We just want to know how adding sprinting is going to affect already peak athletes. And we looked at PDK4. We looked at three genes. PDK4, this is for fat metabolism. VEGF, this is for angiogenesis. This is for creating new blood vessels from old blood vessels. And myostatin MURF1, this is for muscle protein turnover.
Runs into their already prolonged low intensity cycling sessions. And basically, the participants underwent 4 hours of cycling at half of their maximum oxygen level, known as VO2 max. And then they included three sets of 30-second maximal sprints. The measures used were muscle biopsies and blood samples that were taken before, immediately after, and 3 hours post exercise to analyze the changes.
And so this is the genetic analysis of the muscle response. And so we looked at specific primers, which are short DNA sequences, and these are used to target and amplify these genes. Each gene had a pair of primers, one forward and one reverse, and these are designed to bracket the specific segments of DNA they want to examine. This is a common method in molecular biology to measure the abundance of specific mRNA in the tissue. And so the primers were carefully chosen for their efficiency, and these ensure accurate amplification of the genetic material. And for instance, if we look at primer PDK4, they showed an efficiency rate, meaning how well the primers amplified the target DNA. And these measurements are fundamental and they're important for basically comparing the levels of gene expression before and after the cycling sessions and with and without sprints. If we look at the results now, if you're looking at panel A, right? And so we look at mitochondrial function, and this relates to how cells generate energy. And so basically, we saw that exercise protocol adding endurance and sprinting, it didn't have any effect on the specific gene PGC AS1. For angiogenesis, this is for angiogenesis. Basically, we did see an increase in VEGF-A and MURF1. And this is really important for protein breakdown and synthesis, especially in mixed exercise protocol ENS. And so this basically says that indicates that sprints might only be important in the breakdown, but also the potential rebuilding of muscle tissue more effectively than just doing endurance training alone. And so looking at panel C now, we want to look at all genes in as a holistic approach. And so we saw that different genes responded differently to different exercise. And so if you look at the genes related to mitochondrial function, they showed less activation under the ENS protocol compared to just endurance.
In terms of the blood hormone responses, the hormones that were measured were cortisol, GH, IGF-1, testosterone, and SHBG. However, only cortisol and GH had clear response to exercise protocols. That's shown in panel A. Panel B shows that the absolute changes in blood hormone concentration of ENS by calculating the differences. And what's noticed from this is that cortisol and GH, they had a particular spike after the ENS protocol, which is attributed to the additional physiological stress from sprinting. However, there were minimal changes in the other hormones. And ultimately, this provides insight into biochemical responses from this sort of training regimen. Next, isokinetic knee extension torque is used to measure recovery. And so post 3 hours and 24 hours after exercise, they measured knees at the following angles: 60 degrees, 180, and 240 each second. At 60, it showed that they remained fairly consistent after both exercises, ENS and E. However, at 180, there were reductions in torque observed immediately post exercise compared to pre exercise values in both exercise protocols, with the recovery trending by 24 hours. The reductions indicate the impact of fatigue from the exercise on muscle performance at this higher speed. However, um, at 240 degrees per second, there were no significant changes in torque observed at the highest speed. Um, Ultimately, the data across all three panels suggests that muscle fatigue induced by both protocols is most evident at moderate speeds, like 180 degrees per second, where the muscles are likely challenged more significantly than at the lower 60 degree per second or faster 240 degree per second speeds. The recovery of muscle torque to baseline levels by 24 hours post exercise demonstrates good resilience and recovery capacity of the muscle tissues involved.
And so now we're looking at the key findings basically. And so we noticed a notable rise in PDK4 mRNA levels, both immediately and 3 hours after exercise, and also the response to PGC1 alpha. This is a regulator of mitochondrial function. This was less pronounced than we anticipated. This suggests variable impacts on mitochondrial biogenesis and function. This could also mean that while sprints enhance some aspects of cellular adaptation, they might also reduce mitochondrial respiration efficiency. Additionally, typical boosts in gene responses were expected from nutrient depletion. Um, this is also because this might be because carbohydrate intake during exercise might help maintain energy levels but suppress some of the usual stress induced adaptations. Lastly, basically sprint training not only challenges muscles but also influences how effective they use different energy sources and adapt at the cellular level. So looking at mRNA, right, we found that the ENS protocol led to lower, this is cycling with sprints, this led to lower levels of protein called myostatin and higher levels of another called MURF1 right after exercise and few hours after. Lower myostatin is good because it usually means more muscle building. And so basically means more breaking down of older damaged proteins. If you look at angiogenesis, we saw that both protocols led to a higher significantly increased markers of angiogenesis. But the really cool thing about this is that we saw 3 hours after post exercise that there was a lot more robust increase in ENS protocol, which means that by adding sprinting in, we can basically could produce more blood vessels, and so these elite athletes can reach even higher levels of athleticism. If we look at impacts on the ion transfer, this is actually something that was actually unsuspected. We found that after doing sprints, the level of certain molecules that help move ions around in muscles were lower. So this actually might sound like a bad thing, but it's actually a natural response. The body might be temporarily like reducing these levels to help the muscles repair and recover right after intense exercise. So like basically, even though these levels drop shortly after exercise, it doesn't actually mean that the muscle's ability to handle stress worsened. In fact, like sprint training is usually known to improve this ability like over time, over a longitudinal output. And so this suggests that sprints could help cyclists recover better and perform well during high intensity activities in the long run. Lastly, if we look at mRNA turnover, we saw that exercise endurance and sprinting reduces myostatin levels. And so this increases MURF1 which indicate protein breakdown, and also sprinting triggers rapid muscle remodeling. And so, as we said before, um, the big thing to take away from this entire thing, what's the entire purpose of why we did this? We want to look out, as we said before, we mentioned all the results of it, but like, how does it affect you and I? Obviously, we're not elite cyclists, but if elite cyclists can get the effect of it, even like think about it, elite cyclists they're already at such a peak level and they were able to increase their athleticism by adding sprints. Think about you and I, like by our level of athleticism, if we can add in sprints or different types of in interval training, we can also induce higher PGC1 levels in us and different in our different genes that affect those biochemical pathways.